Hello students, welcome to EPG Pathshala. I am Dr. Sweetie Shrimali from Department of Zoology in Acharya Narendra Dev College, University of Delhi. In this lecture, I will be dealing with a module from Immunology. The title of the module is T-cell receptor gene rearrangement and generation of T-cell receptor ligands. After studying this module, you shall be able to understand the difference between antigen receptors of innate and adaptive immune system, appreciate the need for TCRs to mount an immune response against a pathogen, understand how the TCR genes were discovered and characterized, describe the organization of TCR genes, understand the concept and benefits of combinatorial diversity, appreciate the differences between the T-cell receptor TCR or B-cell receptor BCR, describe the stages involved in T-cell development, understand the molecular mechanisms involved in the rearrangement of TCR genes, appreciate the role of various cis-acting and trans-acting elements in TCR gene rearrangement, and understand how the ligands are prepared for recognition by the TCR. The adaptive or acquired immune system combats pathogens with the help of very specialized antigen receptors. The adaptive immune system can provide long-term immunity against a particular pathogen and is very specific in its response. Therefore, it is not surprising that the antigen receptors are also highly specific in their interaction with the antigenic determinants of the pathogens. These antigen receptors are called either BCRs or B-cell receptors or TCRs, T-cell receptors, depending on whether they are found on the surface of B cells or T cells. Like all the other receptors, the antigen receptors also have one domain that recognizes its ligand, or in this case an antigen, and another domain which transduces the signal into the interior of the cell. As would be expected, the antigen recognition domains differ greatly, with the signal transduction domains, while the signal transduction domains show very little variation between different antigen receptors. There are two types of TCRs, TCR alpha beta and TCR gamma delta. TCR alpha beta receptors are composed of two polypeptide chains, an alpha chain and a beta chain. Similarly, TCR gamma delta receptors are also composed of two polypeptide chains, the gamma chain and the delta chain. The T cells bearing TCR alpha beta receptors are much more numerous than those bearing the TCR gamma delta receptor. Further, the repertoire of antigen specificity of the TCR alpha beta receptor is much higher compared to that of the TCR gamma delta. Like the adaptive immune system, the innate immune system also employs receptors to recognize pathogens. These receptors are known as pattern recognition receptors or PRRs. The most widely studied PRR is the toll-like receptor or TLR. Some of the major differences between receptors of adaptive and innate immune system are provided in this table. The first point of difference, obviously, is that the PRRs are the receptors of the innate immune system, while the B-cell receptors and T-cell receptors, or BCRs and TCRs, are receptors of the adaptive immune system. The second point of difference is that PRRs show limited variation in their pathogen specificities and have only few subtypes. The BCRs and TCRs, on the other hand, show extensive variation in their antigen specificities. The third point of difference is that while the PRRs recognize common molecules such as pathogen-associated molecular patterns or PAMPs present on the surface of most pathogens, the BCRs and TCRs recognize specific antigens belonging to a particular pathogen. Since PRRs recognize common molecules on the surface of pathogens, they are not pathogen specific. On the other hand, BCRs and TCRs are antigen specific and can even distinguish between closely related pathogens. The fifth point of difference is that the expression of PRRs does not involve DNA rearrangements, while that of BCRs and TCRs involves DNA rearrangements between genes at the immune receptor loci, for example, immunoglobulin and TCR loci of a TCR specific for a particular antigen by generating monoclonal antibody against it. Let's discuss some of the details of this experiment. First, Kappler and Marrack immunized a mouse, say mouse 1, with an antigen ovalbumin or OVA. After a few days, they isolated T cells from this mouse. These isolated T cells contained a subpopulation of T cells with OVA-specific TCR. These cells are short-lived and therefore, to increase their lifespan, they were fused with cells to form a T cell tumor. The hybrid cells formed after such fusions are known as T cell hybridomas. Then, the clonal population, which was specific for OVA, was injected into another mouse, say mouse 2. After a few days, B cells were isolated from the spleen of mouse 2. Hybridomas were formed from these B cells. Out of these B cell hybridomas, the ones that produced monoclonal antibody against OVA-specific TCR was identified. This monoclonal antibody was then used to precipitate out the OVA-specific TCR from a solution containing all the membrane proteins found on T cells. This technique of using a specific antibody to precipitate out its antigen is known as immunoprecipitation. The importance of this experiment by Kappler and Marrack was that it allowed early immunologists to isolate a TCR specific for a particular antigen, in this case OVA. This seminal experiment facilitated further investigation and characterization of the TCR. In 1984, two research articles were published back-to-back in Scientific journal Nature. Both these landmark discoveries revolutionized the field of immunology. Takmak's group discovered the DNA sequence in the TCR gene in humans, while Davis and Hedrick's group discovered the same in mice. The strategy described by Davis and Hedrick was especially elegant. The biggest hurdle for their experiment was the low expression of mRNA encoding the T cell receptor. They hypothesized that the mRNAs encoding for TCR must be found on membrane-bound polysomes. Thus, by isolating only the membrane-bound polysomal mRNA from the T cell hybridoma, they enriched their sample for mRNA encoding the TCR. The major steps in the experiment are depicted in the flowchart. After enriching their sample for mRNA encoding TCR, Davis and Hedrick prepared P32 labeled cDNA from this enriched mRNA. The enrichment was an important step as it reduced the number of cDNA clones to be screened later. Next, they isolated membrane-bound polysomal mRNA from a B cell hybridoma. This was followed by hybridization of the radiolabeled cDNAs from the T cell hybridoma and mRNA from B cell hybridomas. For the genes expressed in both T cells and B cells, the cDNA and mRNA hybridized together to form double-stranded structures. On the other hand, the cDNA molecules which remained single-stranded were those which are expressed only in T cells. These single-stranded cDNA molecules were cloned. It was hypothesized that any cDNA clone encoding a TCR gene would show a changed DNA profile in T cells compared to cells from other organs. Thus, sudden hybridizations were performed with DNA from various cell types using these cDNA clones as probes. This led to the identification of a cDNA clone which showed different DNA bands in non-T cells and T cells. These results pointed to the fact that the cDNA was formed as a result of DNA rearrangements. Subsequent studies established that the cDNA clone does actually encode for the TCR beta chain. Later, the gene sequences for the TCR alpha, gamma, and delta chains were also discovered using similar strategies. The alpha and beta chains of the TCR alpha beta receptors are encoded by TRA locus and TRB locus, respectively. Similarly, the gamma and delta chains of the TCR gamma delta receptors are encoded by TRG locus and TRD locus, respectively. A common feature of all the chains of the TCR is that they contain a variable region and a constant region. The variable region of the TCR beta and TCR delta chains is encoded by three genes: V, which is short for variable, D, which is short for diversity, and J, which is short for joining, while that of the light chains is composed of only two genes, V and J. On the other hand, the constant region of all the TCR chains are encoded by a single gene, the constant or C gene. As shown in the table, the locations and sizes of the TR loci are different from each other. The TRA locus is about 1000 kilobases in size, is located on the short arm of chromosome 14 and has 43 to 45 V genes, 50 J genes and 1 C gene. The TRB locus is about 620 kilobases in size, is located on the short arm of chromosome 7 and has 40 to 48 V genes, 2 D genes, 12 to 13 J genes and 2 C genes. The TRG locus is about 160 kb in size, is located on the long arm of chromosome 7 and has 4 to 6 V genes, 5 J genes and 2 C genes. The TRD locus is about 530 kb in size, is located on the short arm of chromosome 14 and has 7 to 8 V genes, 3 D genes, 4 J genes and 1 C gene. The number of these genes may vary among different individuals depending on their haplotype. For example, the TRA locus has 43 to 45, TRB locus has 40 to 48, TRG locus has 4 to 6, and TRD locus has 7 to 8 functional V genes. Further, the number of TRBJ genes can be either 12 or 13 depending upon the haplotype of the individual. Besides differing in their location and size, the TR loci differ in their organization also. The figure illustrates the relative positions of the functional V, D, J and C genes. Out of all the loci encoding the immune receptor genes, the organization of the TRA, TRD locus is the most unique. This is because at this locus, the genes comprising the TRA as well as the TRD loci are found. After the V genes at the TRA and TRD loci, the D, J and C genes of TRD locus are present. Although most of the V genes of this locus are present at the 5' end of the locus, the TRD V3 gene is an exception. Its location is downstream or 3' to the TRD C genes. The J and C genes of the TRA locus are found downstream to the TRD V3 gene. The organization of TRB is such that almost all the V genes except one, which is the TRB V30, are clustered together at the 5' end of the locus. The D, J and C genes of the TRB locus form two separate clusters, DJC1 cluster and DJC2 cluster. The location of the TRB V30 gene is peculiar as it's located at the 3' end of the locus. The TRG locus is quite similar in organization to the TRB locus. All the V genes at this locus are located at the 5' end. This is followed by JC1 cluster. The JC2 cluster is located further downstream to the JC1 cluster towards the 3' end of the locus. The double slanting bars in the figure represent the fact that these genes may be hundreds and sometimes thousands of base pairs away from each other. So far, we have discussed the organization and structure of the TR loci at the DNA level. But what of the protein that these loci encode? The protein in question being the T cell receptor or the TCR, which is composed of two polypeptide chains. After its assembly, the TCR is expressed on the surface of T cells. Here it is found in association with the CD3 complex, and the resulting complex is referred to as the TCR-CD3 complex. The CD3 complex is involved in signal transduction and is composed of 6 polypeptide chains which are organized as zeta-zeta homodimer or a zeta-eta heterodimers, gamma epsilon and delta epsilon heterodimers. Although they bear the same names, the gamma and delta chains of CD3 complex differ greatly from gamma and delta chains of the T cell receptor. The polypeptide chains of the CD3 complex contain long cytoplasmic chains that help in signal transduction. So now let's ask ourselves, what is the difference between the T cell receptor and the B cell receptor? Indeed, the BCR and TCR differ greatly from each other in a variety of ways. The first point of difference is obviously that the TCR is found on the surface of T cells, while the BCR is found on the surface of B cells. The second point of difference is that for a TCR, the antigen recognition component is composed of either alpha and beta or gamma and delta chains, while for a BCR, the antigen recognition component is composed of 2 heavy and 2 light chains, which could be kappa or lambda. The third point of difference is that in case of TCR, the signal transduction component is CD3, whereas for the BCR, it is Ig alpha, Ig beta heterodimer. The fourth point of difference is that while the BCR is found in membrane bound as well as secretory form, which is antibody, the TCR is only found in its membrane bound form. The fifth point of difference is that the TCR contains only one antigen recognition site, while the BCR contains 2 identical antigen recognition sites. The sixth point of difference is that the TCR is encoded by genes at the TRA and TRB for TCR alpha beta or TRG and TRD for TCR gamma delta loci. On the other hand, the BCR is encoded by genes at the IgH, IgL and IgK loci. The seventh point of difference is that the TCR recognizes antigen only after it has been processed and presented in the context of a self-MHC molecule, while BCR recognizes antigen by itself. The last point of difference that we are going to talk about today is that the TCR does not undergo somatic hypermutation, so that once a TCR is formed in the thymus, it does not undergo any further change. However, the BCR undergoes somatic hypermutation so that the BCR can change its amino acid sequence after it comes in contact with its antigen. So far, we have discussed that immune loci are composed of multiple genes, but these multiple genes are only 7 to 8% of the vertebrate genome, and therefore, by themselves, they are not enough to encode the vast repertoire of antigen specificity that is found in our lymphocytes. So how is the immune system generating so many antigen specificities? In fact, there are many ways in which the immune system does this. And one of the most important ways is combinatorial diversity. To explain combinatorial diversity, I'll remind you that the antigen binding site of an immune receptor molecule composed of V, D, and J genes. The coming together of these genes can occur in many possible combinations. And this phenomenon of recombination of a few genes such that a large number of antigen specificities can be generated using relatively fewer number of V, D, and J genes is known as combinatorial diversity. By calculating the maximum number of possible ways in which the V, D, and J genes can combine with each other, we can obtain a fairly good estimate of the possible number of antigen specificities that the TR loci are capable of generating. In this table, I am showing you how to calculate the combinatorial diversity of human TR loci. The antigen binding region
of TCR corresponds to V and J genes of the TRA and TRG loci. Therefore, in order to calculate the maximum number of antigen specificities that these loci can generate, we multiply the maximum number of these genes at these loci. For TR locus, 45 multiplied by 50 gives us 2250, while for TRG locus, 6 multiplied by 5 gives us 30. The antigen binding region of TCR corresponds to V, D, and J genes of the TRB and TRD loci. Therefore, to calculate the maximum number of antigen specificities at these loci, we multiply 48 by 2 and 13, which gives us 1248 for TRB locus. For the TRD locus, 8 multiplied by 3 and 4 gives us 96. For calculating the maximum number of antigen specificities for TCR alpha beta, we multiply 2250 by 1248, which gives us 2.81 million. For calculating the maximum number of antigen specificities for TCR gamma delta, we multiply 30 by 96, which gives us 2880. So the TCR gamma delta are much less diverse in their antigen specificity compared to TCR alpha beta receptors. The most striking thing, however, is that due to combinatorial diversity, a very small number of TRA and TRB genes in humans have a potential to encode for TCR alpha beta with a staggering 2.81 million different antigenic specificities. Now that is really something, isn't it? I should point out here that calculating the actual size of the repertoire for antigen specificity is not so simple, because in reality, all the recombination events are not entirely random, and some types of combinations are generated more frequently than the others. Also, other mechanisms besides multiple TR genes and their combinatorial diversity play important roles in determining the variety of antigen specificity. We will discuss all these mechanisms also in a little while. As illustrated in the figure, the TRB and TRD loci contain V, D, and J genes and undergo two separate recombination events. One event links D gene to J genes and is called D to J or DJ recombination. The other event links the rearranged DJ genes to V genes and is known as V to DJ or VDJ recombination. The sequence of recombination events at the TR loci is precise, with DJ recombination occurring before VDJ recombination. Further, the TRB, TRG, and TRD loci get rearranged before the TRA locus. The TRA and TRG loci do not contain D genes and therefore undergo V to J or VJ recombination only. As will be discussed later, the mechanism of molecular processes that lead to all these kinds of recombination events is essentially the same. The precise sequences at which DNA is excised to enable recombination events are highly conserved across most vertebrate species. Further, these sequences are similar between TR and immunoglobulin loci. These sequences are known as recombination signal sequences or RSS. As shown in the figure, an RSS consists of a conserved 7 base pair or heptamer sequence and a 9 base pair or nonamer sequence which flank a relatively non-conserved sequence. Depending upon the length of baser sequence, an RSS can be either a 12 base pair RSS or a 23 base pair RSS. As their name suggests, a 12 base pair RSS contains a 12 base pair long spacer DNA, while a 23 base pair RSS contains a 23 base pair long spacer DNA. An interesting feature of the RSS is that a recombination event can only occur between dissimilar RSS, a phenomenon known as 12-23 rule. Thus, a 12 base pair RSS can only recombine with a 23 base pair RSS and vice versa. Now, since the RSS are the sites where the RAG1 and RAG2 proteins bind the DNA and cleave it. Therefore, they ensure that the somatic recombination occurs at specific sites only. This site-specific nature of recombination is very important as recombination at non-specific sites can lead to cancer. Now that we have discussed the major steps involved in somatic recombination, let us look at the molecular mechanisms underlying this fascinating process. The basic molecular mechanisms underlying all the kinds of somatic recombination are the same irrespective of whether it's D to J recombination, V to DJ recombination, or V to J recombination. The first step in somatic recombination is the binding of the RAG1 and RAG2 proteins to the RSS, which are adjacent to the immunoglobulin genes. After they bind the appropriate RSS, the RAG proteins introduce a single strand break in the DNA between coding sequence and its flanking RSS. This break is introduced at precisely the 5' border of each RSS. Now, as a result of the single-stranded DNA break, a 3' hydroxyl and a 5' phosphate end are generated. Next, the RAG proteins catalyze a transesterification reaction wherein the 3' hydroxyl group at the end of the gene launches a nucleophilic attack on the phosphodiester bond on the opposite non-coding strand. This reaction generates two ends, a coding end with a closed hairpin loop and a blunt signal end. These ends are held together by RAG in a post-cleavage synaptic complex or PSC. Note that overall, two coding joints and two signal ends are generated for each recombination event. The two signal ends generated in the process are joined by DNA ligase 4 to form a signal joint.
Next, the enzyme Artemis cleaves the hairpin loop. This cleavage can be asymmetric or symmetric. Asymmetric cleavage generates sites for p-nucleotide addition, which is called so because the filling in reaction by DNA polymerase generates palindromic sequences. Simultaneously, two more processes occur which, along with p-nucleotide addition, increase the junctional diversity during recombination. One of these processes is the exonuclease-mediated DNA cleavage. The other process is N-nucleotide addition and is called so because it brings about non-templated addition of random nucleotides. The introduction of nucleotide variations by p-nucleotide addition, exonuclease cleavage, and N-nucleotide addition at the junction between two genes during somatic recombination is known as junctional diversity or junctional flexibility. Remember that junctional diversity is different from combinatorial diversity. The next step involves filling in of the gaps by DNA polymerase. Finally, the coding ends are ligated to form a coding joint with the help of DNA ligase 4 and non-homologous end joining proteins. In case of VJ and VDJ recombination, this stage represents the end of somatic recombination. However, in case of DJ recombination, this stage is followed by reinitiation of recombination to link a recombined DJ to a V gene, that is VDJ recombination. We have already discussed that the somatic recombination is a developmental stage-specific process. All the lymphocytes are derived from the common lymphoid progenitor or CLP. The development of T cells occurs in the thymus. The stage of T cell development has profound effects on the process of TCR gene rearrangement. The developmental stages of T cells are studied with respect to the expression of certain well-defined surface molecules. These surface molecules are usually designated CD, which stands for cluster of differentiation. CD4 and CD8 are among the most widely studied T cell surface marker molecules. Depending on the presence or absence of CD4 and CD8 on their surface, the T cell developmental stages are defined as double negative or DN, which does not have either CD4 or CD8, double positive or DP, which have both CD4 and CD8, and single positive or SP, which either have CD4 or CD8. Of these three stages, the DN stage is the earliest stage during T cell development. Based on the presence or absence of two more cell surface molecules, CD25 and CD44, the DN stage can be further subdivided into DN1, DN2, DN3 and DN4 stages. DN1 is CD44 positive, DN2 is positive for both CD44 and CD25, DN3 is positive only for CD25 and DN4 is negative for both CD44 and CD25 stages. The rearrangement of the TRB, TRG and TRD genes occurs at DN2 or DN3 stages. Therefore, this is the time at which T cell fate decision occurs between TCR alpha beta and TCR gamma delta lineages. If the TRG and TRD loci get rearranged, then the cell gets committed to TCR gamma delta lineage. However, if a successful rearrangement occurs at the TRB locus, then the cell gets committed to TCR alpha beta lineage and combines with a pre-T alpha chain to express a pre-T cell receptor or pre-TCR on its surface. The expression of pre-TCR leads to alleelic exclusion so that the other TRB allele does not rearrange a different TCR beta chain. The TCR alpha beta lineage cell then reaches the DN4 stage and starts rearranging the TRA locus. The TCR alpha chain then combines with the TCR beta chain, thereby expressing a T cell receptor on its surface when it reaches the double positive stage. Later, the T cell undergoes positive and negative selection and finally enters the peripheral circulation. Many factors affect the process of TCR gene rearrangements. Some of these factors have to be present on the same DNA to have an effect and are known as cis-acting factors. On the other hand, there are many factors which do not need to be present on the same DNA to have an effect. Such factors are said to exert their effect in trans and thus are known as trans-acting factors. Examples of cis-acting factors are DNA elements such as enhancers, promoters, RSS, while trans-acting factors include proteins such as RAG1 and 2, TDT, RTMS, XRCC, DNA ligase 4, non-homologous end-joining proteins, high mobility group proteins or HMG1 and 2, and Ku proteins. The TCR recognizes its antigen only after it has undergone the necessary processing and is presented with a self-MHC molecule. Thus, it can be said that in case of T cells, both the receptor as well as its ligand have to be generated in a specific manner. This is in absolute contrast with antigen recognition by BCR, which recognizes the antigen directly as it is. The single positive or SP T cells can be either CD4 positive, which is the T helper cells, or CD8 positive, which is the cytotoxic T cells. The TCR of a CD4 positive T cell recognizes exogenous antigens, right? And these antigens are processed and presented in the context of class 2 MHC molecule. Now, class 2 MHC molecules are expressed only in antigen presenting cells or APCs. These APCs internalize the exogenous antigen via endocytosis or phagocytosis and then process it by breaking it down into smaller peptides. These small peptides are then loaded onto the class 2 MHC molecule and subsequently are expressed on the surface of the APC. The TCR on the surface of CD4 positive T cell interacts with this peptide class 2 MHC complex and in combination with appropriate co-stimulatory signals activates the CD4 positive T helper cells. The TCR of a CD8 positive T cell, on the other hand, recognizes endogenous antigens which are processed and presented in context of class 1 MHC molecule. The class 1 MHC molecules are expressed in all the nucleated cells. The endogenous antigen is processed by altered self cells or ASCs, which may either be virus infected or cancerous cells. In the cytoplasm, the endogenous antigen is broken down into smaller peptides which are then loaded onto the MHC class I molecule present in the endoplasmic reticulum. This peptide class I MHC complex is then expressed on the surface of the altered cell. The TCR on the surface of CD8 positive T cells interacts with this peptide class I MHC complex and in combination with appropriate co-stimulatory signals activates the CD8 positive T cytotoxic cells to become cytotoxic T lymphocytes. So students, let us summarize what we have learned in this module. The first thing that we've learned is that TCR alpha, beta, gamma and delta chains are encoded by TRA, TRB, TRG and TRD loci respectively. The variable region of the TCR beta and delta chain is encoded by 3 genes V, D and J genes. The variable regions of TCR alpha and gamma are encoded by only 2 genes V and J genes. On the surface of T cells, the TCR is found in association with a CD3 complex and the resulting complex is known as TCR-CD3 complex. The CD3 complex is involved in signal transduction and is composed of 6 polypeptide chains. The T lymphocytes and B lymphocytes use the same molecular machinery to rearrange the TR and the IG loci respectively. The TRB locus rearranges during the double negative 2 and double negative 3, that is DN2 and DN3 stages, while TRA locus rearranges during the double positive stage of T lymphocyte development. Now, RAG genes are essential for the process of somatic recombination and their deficiency results in immunodeficiencies of both B and T cells. RSS or recombination signal sequences are those DNA sequences where the RAG proteins or RAG proteins bind the DNA to cleave it during somatic recombination. We also learned that TCR recognizes its antigen only after it has undergone the necessary processing and is presented with a self-MHC molecule. And last but not the least, the diversity of the immune system receptor is brought about by the fact that there are multiple genes, the fact that there is a combinatorial diversity for these genes, and junctional diversity, which is brought about by N-nucleotide addition, P-nucleotide addition, and exonuclease trimming. With this, I would like to conclude this chapter. Thank you.